Hello friends, welcome to my video. In this video, we are going to see about pluripotent stem cells. Pluripotent stem cells arise from totipotent stem cells. Pluripotent stem cells can self-renew and differentiate into any of the three germ layers, which are ectoderm, endoderm, and mesoderm. These three germ layers further differentiate to form all tissues and organs within a human being. One point should be noted here, that is, human pluripotent stem cells can give rise to all of the cells of the body but cannot generate a placenta. Embryonic stem cells are the best example of the pluripotent stem cells. Embryonic stem cells are unique cells that exist in an early stage embryo. Before seeing about embryonic stem cells, we'll see about blastocyst. Fertilization is the process in which haploid gametes fuse to form a diploid single cell called zygote, which undergoes rapid cell division to form the blastula. The blastula is usually a spherical layer of cells surrounding a fluid-filled or yolk-filled cavity. Mammals at this stage form a structure called the blastocyst. The blastocyst possesses an inner cell mass which subsequently forms the embryo. The outer layer of the blastocyst consists of cells collectively called the trophoblast. The inner cell mass consists of embryonic stem cells that will differentiate into the different cell types needed by the organism. Human embryos reach the blastocyst stage 4 to 5 days post-fertilization, at which time they consist of 150 cells. Let us see the definition and other important details of the embryonic stem cells. Embryonic stem cells are stem cells derived from the undifferentiated inner cell mass of a blastocyst, an early stage embryo. These cells are pluripotent, that have the capacity to self-renew by dividing and to develop into the three primary germ layers of the early embryo and therefore into all cells of the adult body. Embryonic stem cells are isolated from very young mammalian embryos. Unlike adult stem cells, embryonic stem cells can be cultured indefinitely. Human embryonic cells have been isolated from embryos provided by in vitro fertilization clinics. Worldwide, dozens of genetically distinct human embryo stem cell lines, each derived from a single embryo, are available for experimental investigation. The long-range goal of clinical researchers is to learn how to coax embryonic stem cells to differentiate in culture into desired cell types that can be used for cell replacement therapy. Friends, I am concluding this video here and see you soon in my next video. Thank you, friends.